Hi guys, and welcome to the second video in the series of COVID-19 disease. In this video we'll be looking at the COVID-19 pathogenesis, and just to start off, let me say that the COVID-19 pathogenesis is intimately linked to the renin-angiotensin-aldosterone system you might have heard of. So we will begin by looking at the renin-angiotensin-aldosterone system. Angiotensinogen is synthesized by the liver, and in the presence of decreased pressure, decreased sodium, or increased sympathetic tone, the kidneys produce the enzyme known as renin. Renin is involved in the conversion of angiotensinogen to angiotensin I. The lungs contain an enzyme called angiotensin converting enzyme, or ACE for short. The angiotensin converting enzyme converts the angiotensin I to angiotensin II. Angiotensin II has multiple effects. In the heart, it causes muscle hypertrophy. In the brain, there is increased sympathetic outflow. In the blood vessel, it causes vasoconstriction. In the adrenals, it causes aldosterone secretion. And in the kidneys, it causes increased sodium and water retention. Now this is the classic renin-angiotensin-aldosterone pathway. There is also an enzyme known as ACE2, or angiotensin converting enzyme 2, which converts the angiotensin II to angiotensin 1,7. This angiotensin converting enzyme 2, or ACE2, is the target for the SARS CoV2, which is responsible for the COVID-19 disease. The classical renin-angiotensin-aldosterone pathway ends in angiotensin II, which acts on the AT1 receptor and causes vasoconstriction, increased BP, inflammatory response, prothrombotic response, and sodium reabsorption. The alternative RAS pathway we have discussed here ends in angiotensin 1,7, which acts on the MAS receptor, leading to vasodilation, reduced BP, anti-inflammatory response, antithrombotic response, and sodium excretion. So angiotensinogen gets converted by renin to angiotensin I, which gets converted to angiotensin II by angiotensin converting enzyme. The angiotensin II acts on the AT1 receptor, leading to increased inflammation, increased BP, and increased organ damage. Now this is the classic RAS pathway. In the alternate pathway, the angiotensin II gets acted on by the ACE2 enzyme on the cell membrane, as shown here, which converts the angiotensin II to angiotensin 1,7, which acts on the MAS receptor, leading to reduction in inflammation, reduced BP, and reduced organ damage. As you can see, the alternative RAS pathway we have discussed here, ending in angiotensin 1,7, acting on the MAS receptors, causes reduced inflammation, reduced BP, and reduced organ damage, whereas angiotensin II in the classic RAS pathway, acting on the AT1 receptor, leads to increased inflammation, increased BP, and increased organ damage. Now, the spike protein of the coronavirus attaches to the ACE2 enzyme on the cell membrane, and this attachment to ACE2 enzyme, as shown here, means that the conversion of angiotensin II to angiotensin 1,7 does not occur. Thus, the alternate RAS pathway of angiotensin 1,7, leading to reduction in inflammation, reduced BP, and reduced organ damage, does not occur. And the classic RAS pathway predominates, leading to increased inflammation and increased organ damage. Now let us look at how the virus particles attach to the ACE2 receptor and how the virus particle replicates itself. So shown here is the COVID-19, the SARS CoV2 particle itself. marked here is the S protein, the ACE2 enzyme present on the cell membrane, and the cell membrane. The spike protein attaches to the ACE2 enzyme, and through endosomes it's taken into the human cell. This process is known as fusion. Now after fusion, proteolytic enzymes like cathepsin L acting in an acidic medium causes breakdown of the viral protein, and this is a target for the drug chloroquine or hydroxychloroquine. We will discuss specific actions of drugs in a later video. Once the proteolytic enzymes are acted on, the viral RNA is released into the human cell, and the RNA-dependent RNA polymerase is synthesized, which leads to copying of the viral RNA material. Now this step is a potential target for the drug favipiravir and penciclovir, and also a target for remdesivir. As we already said, the RNA-dependent RNA polymerase is involved in creating multiple copies of the viral genetic material, and the virus uses the human rough endoplasmic reticulum to synthesize viral proteins. These viral proteins are reassembled and taken out of the cell, and the virus particles are released to the environment. This is a potential target for oseltamivir. Now, once the virus particles are released, the other cells of the body may also get involved, and the process carries on. Now, angiotensin converting enzyme 2 is expressed in the lung, the kidneys, the heart, the intestines, brain. Now, this may explain the symptom complex found in COVID-19, which we will talk about in a later video. We will also try to discuss regarding the symptom complex, the presentation types, the prognostic factors, and also treatment options in a later video. Let us stop for now. Thank you for watching my video, and please don't forget to subscribe and ring that notification bell so you can be notified of further videos, and also don't forget to like and comment on the video. As always, I'll be leaving links in the description box below. You can use the links to read up further on the topic we have discussed. Hoping to see you in the next video. Thank you.